Let's get started. And just like any good fortress, you've got to start with the outer defenses. A bacterium's number one job is to protect itself, so that's where we'll begin our tour. So that very first line of defense for a lot of bacteria is something called the capsule. You can think of it like this slimy biological invisibility cloak. Seriously, this layer helps it hide from your immune system, and it makes it super slippery, so it's tough for other cells to grab onto and gobble it up. It is just incredibly effective. Okay, so right under that slimy capsule, we hit the main wall of the fortress, the cell wall. Now, this isn't just a floppy bag. It's this super rigid structure that gives the bacterium its shape and, crucially, keeps it from basically exploding from internal pressure. And the building material? It's this really special stuff called peptidoglycan. And this is where things get really interesting. See, other organisms have cell walls, like plants and fungi, but theirs are made of stuff like cellulose or chitin. But bacteria? Nope, they use peptidoglycan. And that unique choice of building material, well, it sets them apart from pretty much all other life, and especially from our own cells. So you might be thinking, okay, different building materials, so what? Well, here's the kicker. That unique material is both its greatest strength and its greatest weakness. It's the exact target for some of our most powerful antibiotics. You've heard of it, penicillin. By attacking the peptidoglycan, we can literally tear down the fortress wall without even touching our own cells. All right, one more layer to go. We've made it to the inner gatekeeper, the cell membrane. And there are a couple of really fascinating things about it. First, unlike our cells, it doesn't have stuff like cholesterol. But second, and this is a total game changer, it is packed with respiratory enzymes. What does that mean? It means the membrane itself is a little powerhouse for energy production, a job that our cells need whole separate compartments for. Okay, so the fortress is secure, the defenses are up, but hey, you can't just sit in your fortress all day, right? A survivor needs tools to move around, to grab onto things, and to interact with its world. So now, let's check out the external gadgets. First up, motion. How does a little bacterium zip around? Well, say hello to the flagella. These things are basically incredible little biological outboard motors. They're made of a protein called flagellin, and these long, whip-like tails spin around just like a propeller, pushing the cell right through whatever liquid it's in. Okay, so movement is covered, but sometimes you need to stay put. And for that, bacteria have pili. These are thinner, shorter little filaments, and they work just like tiny grappling hooks. They let the cell cling to surfaces, and yeah, that includes the tissues inside a host. But that's not all they do. They have another, much more complex job. So, job number one is just attachment. Simple, but absolutely key for setting up shop. But the second job, conjugation, this is just mind-blowing. One bacterium can use a special pilus to form a direct bridge to another bacterium and literally pass along chunks of genetic information. It's how bacteria share secrets, passing on genetic code to their neighbors and adapting at lightning speed. All right, we've officially breached the outer walls. We've seen the tools on the outside. Now it's time to step inside the engine room itself, the cytoplasm. This is where all the real action happens. So you look inside, and one of the first things you might notice is the mesosome. And it's a really clever trick. See, bacteria don't have all the fancy internal compartments that our cells do. So what do? They just fold their cell membrane inward over and over again. This massively increases the surface area for really important jobs, like respiration or helping to build a new wall when the cell is ready to divide. And scattered all through the cytoplasm, you'll find thousands of these little protein-building factories called ribosomes. Now, bacterial ribosomes are a little smaller than the ones in our cells. We call them 70S ribosomes. And guess what? That size difference is yet again another perfect target. Some antibiotics are designed to only shut down these 70S ribosomes, which stops the bacteria from building the proteins they need to survive. Okay, now we're getting to the absolute heart of it all, the real secret sauce, the core of what makes bacteria the ultimate survivors. It's not just their walls or their motors, it's their incredibly slick and efficient genetic toolkit. You know, our cells are so complex, we package a ton of DNA into a lockdown nucleus. Bacteria, they keep it simple, they keep it fast. Their entire main blueprint is just a single circular chromosome of DNA hanging out right there in the cytoplasm. No nucleus, no waiting around. The info is right there, ready to go for super fast growth and response. But wait, there's more. Let's talk about plasmids. Think of these as swappable software plugins. They're these little extra circles of DNA that carry bonus genes. And this is where it gets really serious for us. A single one of these plasmids can carry the genes for resistance to multiple different antibiotics. So a bacterium can grab one of these through that conjugation bridge we talked about and in an instant go from treatable to a full-blown superbug. Okay, so if plasmids are plugins, then transposons, they're the wild cards. We're talking about jumping genes. These are literally segments of DNA that can just decide to cut themselves out of one spot on the genome and paste themselves somewhere else. This constant shuffling of the genetic deck, well, it's a crazy powerful engine for rapid-fire evolution. So let's just recap this amazing genetic toolkit. You've got your core operating system, the chromosome. Then you have these swappable upgrade modules, the plasmids, bringing in new features like resistance. And finally, you have the transposons, which are constantly shuffling and moving genes around, creating new combinations. This three-part system, it's what makes bacteria so unbelievably adaptable. So when you take a step back and look at the whole picture, the fortress-like defenses, the simple but super effective tools, and this just incredibly dynamic genetic toolkit, you realize you're looking at a masterpiece of survival. And it leaves you with a pretty wild thought, doesn't it? For all of our complexity, with their simple design and their absolute mastery of adaptation, could it be that these tiny cells are the true rulers of our planet?